All right, this is a review of transcription regulation. This is a figure from your book. This is figure 8.3. It's in the 8th chapter, and we'll talk about why our author uses it there and why we use it right at the start of our course. This is considered a review of transcription regulation, but we'll take what you know and add a lot to it. This goes along with a handout that's given in class. It's available on the course website. And so what we're looking at this figure is any gene. It's any gene in any eukaryotic organism. The story is really similar in prokaryotic organisms, but the terms are different. And so we're really focused on eukaryotic organisms, but we're focused on any eukaryotic organism like any plant or animal. And any gene, we'll talk about what a gene is. A gene is DNA. And so the first thing to look at in this figure, as you see here, there you can see that's the double-stranded DNA. So this is the genetic code, the AGCTs that you inherited from your mom and dad. It's DNA, it's the DNA double helix, and this sort of piece of cooked spaghetti is DNA. So we're looking at DNA, and we're looking at the DNA of a gene, and this could be any gene, but we'll use in our example, we'll say it's the insulin gene. And the insulin gene is just made up of DNA. We'll get to the other objects in this figure in a couple of minutes. These are proteins that all the colored objects are proteins, and we'll introduce them. But to start out with, we're going to focus on the fact that this DNA is a gene, an insulin gene, and we have this gene in every cell in our body. And so that's one of the key concepts in developmental biology of plants and animals, is that in general, with very rare exceptions, plants and animals have all of the genes for the organism in every one of their cells. So for example, we have the insulin gene in our brain cells, even though it's not used there. A big part of the story of developmental biology is using a subset of genes in all the different cell types. So the insulin gene, for example, is only used in some of the cells in our pancreas, and it's present in all the other cells, but it's not used. One of the key ideas there is that this DNA is exactly the same in all those cells. So we're going to talk about the organization of the DNA, but the difference between the pancreas cells and the brain cells are these proteins that are going to be bound to the gene. So it's going to be the DNA first, and the DNA is the insulin gene, and the insulin gene is broken up into two parts. And there are two parts for any gene in any living cell. In eukaryotes, we can talk about these two parts as being called this part of the DNA, which is called the transcription unit, and this part, which in this course, developmental biologists typically call that whole region the promoter. And the handout that goes along with this helps sort of clarify some of the terminology. Your author calls this the promoter, but in our course, most developmental biologists would call this the basal promoter. And we'll talk, that's a good term. It means sort of the basic or minimal promoter. You'll also see in some of the reading that you do in your papers for this course, it might be called the proximal promoter, which is also a good name because it's proximal, it's right next to the transcription unit. So two parts to any gene, the transcription unit and the promoter. We'll first talk about the transcription unit. The transcription unit is the part of the gene that gets copied. The transcription unit gets copied into RNA. And that first copy into RNA that gets made is called the primary transcript. And the primary transcript gets spliced to form mature RNA for the case of, say, a protein coding gene, mRNA. Oops. But this is true for any gene. So we also have genes that are getting spliced to make ribosomal RNA and transfer RNAs and microRNAs, like that, a variety of different RNAs. We usually will think about mRNAs, and we'll think about mRNAs that are going to code for a protein. That's true in this case. The insulin gene has a transcription unit that includes the coding region for the insulin promoter protein. And what happens there is that the insulin gene in the pancreatic cells gets copied into RNA, and inside the nucleus, the primary transcript gets spliced or processed into mRNA, and the mRNA goes out into the cytoplasm of the pancreatic cells and gets translated into insulin protein. So the transcription unit is could be called sort of the business part of any gene. It's the part that does the job of the gene. And we'll look at this part in detail in our class. Again, this is the part that includes the information to code for the insulin protein. The other part of the gene is called the promoter, and we'll just take a look at that here. And so I'll just look at this second part of the promoter like that. And we've already said that the promoter has something called the basal or proximal promoter. That's here. And so we can draw this line here and say every promoter is composed of two parts. One part is the basal promoter. The basal promoter is always next to the start of transcription. In other words, it's right next to the transcription unit. So here's the basal promoter right next to the transcription unit of the insulin gene. The other part of the promoter is here, and almost all scientists refer to the other part of the promoter as enhancers. But that's a poor word because, and especially true in developmental biology, this part of the promoter has both enhancers and suppressors. And so we want to call this, we might call it the distal promoter. And the distal promoter is far away from the gene. This region here could be long distance. It could be thousands of nucleotides. I should have mentioned the overall length of a typical gene just to give you a feeling. We're talking about AGCTs as being the nucleotides, and genes vary greatly in length. Some of the shortest genes would be only maybe 1,000 letters long. Some of the longest genes, like say for example the cystic fibrosis gene, is 250,000 letters long. And lots of this space that's filled up in long genes is just big gaps. So for example, this part of the distal promoter is just worthless space. The key parts of the distal promoter are these purple elements, which are called enhancers here. A better word for that is, from our handout, is developmental biologists often call these response elements. And that's because they can be either enhancers or suppressors. And sometimes one response element can be an enhancer at some points in time or in some cell types, and in another cell type or point in time in development it can be a suppressor. So again, all biologists call these enhancers, but that's just not that great a word, and we'll hear these other words like response elements in there. Again, we're still talking about the DNA. So for example, the insulin gene has the same response elements in every cell type. Okay, we'll find out that response elements are a really important part of the gene, but in the case of genes, they're always the same in every cell type. Okay, so we talked about the transcription unit as being the business end of the gene. The promoter is the regulatory end of a gene. And the promoter tells where, when, and how much of the gene to transcribe. Okay, so we have a transcription unit that's going to code for, in our case, the insulin mRNA, and the promoter is the part of the gene that says make this gene only in the pancreas and make it when the cells have matured and the organism's ready to have insulin function, and then make lots of it, because these are specialized cells that need to produce insulin that's going to spread through the bloodstream to the entire body. So that's what the promoter is, and again, that's all the DNA all about the DNA in this story. I'm going to stop here and make a second part to this where I describe the colored objects.